The production of a protein is like the production of a car, with the protein being made up of different amino acids, similar to how a car is made up of different parts. Additionally, every amino acid is translated from a group of 3 RNA nucleotides, similar to how every part of a car may be made up of different subcomponents. We are given a 30 base pair long sequence of double-stranded DNA. To understand what protein these strands code for, we have to first locate the group of 3 nucleotides that, when transcripted and translated, code for the amino acid methionine, also known as the start codon. Methionine is like the first and most essential part of the car. It can be seen that the bottom strand includes the sequence ATG, which when transcripted into mRNA and translated codes for methionine and therefore is the coding strand. When the RNA strand is completely translated, the amino acid that build up the protein are methionine, valine, arginine, phenylalanine, and lysine. These are like the engine, body, window, doors, and tires that are needed to produce a car. Many mutations can happen in the DNA, which ultimately affects the process and the production of proteins. Transition mutation takes place when a single nucleotide is swapped with another nucleotide of the same category, whether it is swapping of A and G, which are both purines, or CNT, which are both pyrimidines. A transversion mutation happens when a single nucleotide is accidentally swapped with a nucleotide from another group, and the purine is misplaced for a pyrimidine or vice versa. A mutation is said to be silent when a nucleotide is swapped with another nucleotide, but the mutated codon codes for the same amino acid when transcripted and translated, while the rest of the amino acid sequence is not impacted. This is like using 2 different spellings to pronounce the same word. Kourosh and Kourosh are spelled differently, but both sound the same, and are different spellings for the same name. A missense mutation happens when a point mutation takes place and the resulting 3 base pair codon codes for a different amino acid in a protein. This is similar to how changing a letter in a word can completely change the meaning of the word. When a point mutation occurs, where after transcription and translation a 3 base group codon that is not supposed to codes for a stop codon, it is said that the mutation is a nonsense mutation. This results in decreasing the number of amino acids present in the protein. This is similar to cutting the end of a word to come up with a new word. Lastly, a read-through mutation takes place when a point mutation happens at a stop codon, changing it to a codon that codes for an amino acid after transcription and translation, which results in a longer protein. This is similar to extending a word and adding more characters to it to end up with a new word.